So I can share my screen.
It goes around sometimes to a couple other departments. So we're really pleased and excited to have Kristin Schill with us today. She's going to speak to us about the continued search for suitable surrogates for Clostridium botulinum. Kristin is one of our own, we'll call her. She was here with us for how many years, Kristin? I don't know how long you were here first.
10.
10 years, okay.
Yep.
10, okay, so 10 years at iFish working on the FDA side, and then she was swooped up by University of Wisconsin. They certainly got a phenomenal scientist, and we miss her a lot. There was kind of a bio that you can read through in detail, as well as her abstract. And so without taking any more time, help me welcome Kristin to our seminar series.
Thanks, Britt. And I miss you guys. I miss everybody there. So yeah, thanks for the introduction. It's really a pleasure for me to actually give a talk to you about this very important topic for food safety, and that's the continued search for suitable surrogates. And I say continued because we still don't have suitable surrogates for growth inhibition studies for Clostridium botulinum. We have thermal surrogate as for group 1 strains, as I'll mention later on in the talk, but no, we don't really have suitable surrogates for growth inhibition studies. And actually, I've prepared some slides to give you an idea of more of my background. Like I said, I gear this more towards students, but I've actually been working on surrogates ever since I was a master's student. Switch to the next slide. Okay. So I went to Iowa State University and worked on my degree in microbiology, my bachelor's degree in microbiology, and my master's in food science. And I worked with Jim Dixon in his laboratory, his food safety research laboratory. And my project at that time was looking for indicators or surrogate organisms for E. coli 0157H7 for fresh meat processing. So we had isolated a variety of gram-positive and gram-negative bacteria, and we examined the response to high temperature and pH conditions, different pH conditions. And then we wanted to see what their response would be to various carcass wash treatments and how that compared with the carcasses are often washed with hot water, cold water, organic acid washes, or other sorts of carcass wash intervention methods to eliminate pathogenic microbes. And we had found 5 generic E. coli isolates that behave similarly as 0157H7. So when the various carcass wash treatments were applied. So we suggested their use as indicators or surrogates for 0157H7. And then these isolates were deposited into the American type culture collection, and they continue to be used today to validate 0157H7 and salmonella inactivation strategies for fresh meat processing. So I moved to UW-Madison to pursue a career or a PhD in food science, and I worked with Eric Johnson in his laboratory, and that's where I was introduced to Clostridium botulinum. So the first project I worked was to determine whether the nematode, Cenorhabditis elegans, could serve as an alternative to the mouse bioassay. But after dumping boatloads of toxin onto these nematodes, they just kept swimming around. So we looked at whether or not, are they taking up the toxin, or because they eat by siphoning. So we thought, well, we could try microinjection, but that's really expensive. And the place on campus did not want us to bring the most potent toxin on the planet into their genetics lab, where they were walking around inside the lab with cups of coffee. So they were not too thrilled. So then zebrafish was another suggested model by one of the committee members. But if you think about it, you're going to have these 10-gallon fish tanks that you're going to be inoculating with botulinum neurotoxin. And that just did not seem safe. So we actually trashed that idea and went on to toxin regulation. And I should point out that there still is not an alternative to the mouse bioassay. So I don't know if there will be anytime soon, but for the past several decades, there hasn't been. So anyway, so my dissertation, ended up centering on toxin regulation, which we still don't know a lot about for Clostridium botulinum. But we found that in certain C. botulinum strains that produce small amounts of toxin, the toxin genes were located on these very large plasmids in certain strains. And then we used a Clostridium mutagenesis tool to site-specifically inactivate the toxin genes that were encoded on these plasmids as a means to track the plasmid movement into different strains to see if they could move and whether or not they were conjugative plasmids or mobilizable by some other mechanism. So also using this Clostridium tool, we were able to construct a mutant of the serotype A strain 62A, which we had proposed could be used in food challenge studies as a surrogate for Clostridium botulinum. However, Clostridium is a site-specific inactivation tool, and I'll go into this in a little bit more detail, but it doesn't produce a complete toxin gene deletion. So if the intron is excised out, then there could be a restoration of the toxin gene. And so that strain, that mutant that we developed, really hasn't been utilized by anybody in industry or any other group. It was not found to be, or not sought after by petitioning to CDC to become an exempt strain or a non-toxigenic strain. So then I left Madison and came to work with the FDA at Moffitt, as most of you know, and worked with a lot of you. And I was there for 10 years and worked in the C. botulinum laboratory as a co-PI in the Clostridium botulinum lab, or as the Honorable N. Rukma Reddy laboratory. And Guy remembers there was a sign posted outside the lab. That's what it was called after one of the other PIs. So we worked on a lot of different Clostridium botulinum projects that centered on looking at developing and validating toxin detection assays, looking at the thermal resistance and the resistance of C. botulinum spores to high pressure processing. We looked at genetic characterization of C. botulinums and C. sporogenies. C. sporogenies is another closely related species to group 1 C. botulinum. And that's this group of organisms, C. sporogenies, is going to be play another role later on in the talk. And I'll talk about that in more detail. But we also looked at a little bit on toxin regulation. So in January of 2020, right before the pandemic, I left FDA and came up to the Food Research Institute, where I helped lead the Applied Food Safety Laboratory. And we work with the food industry to design and conduct food challenge studies. And we design these challenge studies on a variety of foodborne pathogens that I've listed here. So not just BOT, but your gram positives and gram negatives. And then we also work with a lot of different food products. So it keeps us on our toes for sure. So a little bit about the Food Research Institute. For those who don't know, the FRI has been in existence since 1946. And we facilitate and coordinate all the food safety research here at UW-Madison. So we collaborate with the food industry, which produces foods regulated by both USDA and FDA. So we cooperate with those regulatory agencies. And there are affiliated UW faculty members that belong to the different departments and colleges on campus, such as bacteriology, the food science department, medical microbiology and immunology department, the meat science and animal biologics discovery, and the Wisconsin Vet Diagnostic Laboratory, just to name a few. So we're an interdepartmental collaboration, much like iFish. But because we're an academic institute similar to iFish, we train undergraduates and graduate students and postdoctoral fellows. So part of our mission is to be a source of well-trained scientists in the field of food safety. And one of the most important missions that we have is through our outreach by giving seminars and webinars to our sponsor companies. So this is a listing of some of our research objectives at FRI, and they include pre-harvest and post-harvest interventions. Again, food challenge studies, which encompasses a lot of our applied food safety research. We have experts that focus on the mechanisms of pathogenesis as well as virulence of various foodborne pathogens. Dr. Kathy Glass has been the lead of the Applied Food Safety Laboratory for the past 35, 36 years now, and she's really built up a fantastic team of researchers who conduct these food challenge studies for the different companies. And we look at a variety of different foods, again, dairy, meat, bakery, refrigerated foods, and a significant portion of these studies involve pasteurized processed cheese products and the use of antimicrobials, both traditional and natural or clean label, to control pathogen growth in foods. We're recently encountering a lot of projects focusing on the safety of plant-based protein foods. So there's always something new or different that we are working with, and it really does keep us busy and constantly studying new concepts. And conducting these food challenge studies helps companies validate their processing strategies or their food formulations. So we help to identify novel strategies and formulations to improve the safety. So now that you know a little bit about me and a little bit about the Food Research Institute, I'd like to move on to the discussion regarding surrogates for Clostridium botulinum. But then this requires a little bit of background of what Clostridium botulinum is for some of those of you who don't know. So historically, if a strain produced botulinum neurotoxins, it was named Clostridium botulinum, which was a species that was broken down into 4 different groups. But now they're broken into, groups 5 and 6 have been added to represent Clostridium botulinum and Butyricum. But these strains were broken down into these different groups based on their metabolic, genetic, and physiological differences. As new strain subtyping mechanisms have been developed, such as whole genome sequencing, we've been able to more accurately identify the taxonomy of this heterogeneous group. Neurotoxigenic Clostridia are gram-positive, spore-forming anaerobic bacilli, meaning they don't like oxygen. And because they form resistant endospores, they can be found virtually everywhere in the environment. So they also produce 7 confirmed serotypes of botulinum neurotoxins, represented at A through G. And I've shown these listed here. Rare strains of Clostridium botulinum and Clostridium Butyricum have been isolated that produce type F and type E neurotoxins respectively. And the genes that encode for botulinum neurotoxins and these associated proteins, they're found within a gene cluster, and these are been located to mobile genetic plasmids, bacteriophages, and other mobile genetic elements that permit its sporozenal transfer. And this helps to give rise to these variety of strains that produce these toxins. Group 1, Clostridium sporogenes, was thought to be a non-toxigenic counterpart, and typically it is, but there have been strains of Clostridium sporogenes that have been found to carry the type B toxin genes. And so for this presentation, we'll only focus on groups 1 and 2, because these are the strains that cause botulism in humans. And group 1 represents the proteolytic strains, which means that they can produce the enzymes that catalytically activate the toxin into an active dye chain molecule, as well as degrading proteins, whereas the non-proteolytic strains have to rely on enzymes within the gut of the host or within the food product to activate the toxin molecule. The group 1 strains have higher optimum growth temperatures, and they form spores that are more heat resistant than the group 2 strains. The group 2 strains actually are a concern for refrigerated extended shelf life foods because they can grow at refrigeration temperatures. So as I mentioned, they produce 7 confirmed serotypes of botulinum neurotoxins. Types A, B, E, and F cause botulism in humans, whereas type C and D produce symptoms of botulism in animals. And type G has not yet been associated with causing botulism in humans. But this is so there's different types of botulism. There's foodborne botulism. This is wound botulism. I don't know if you can see my cursor, but I'm pointing to this top picture here. But you can see that this is a form of wound botulism, and this is botulism in animals, where this causes the limber neck or the paralysis of the disease. I also want to point out that there's dual neurotoxin producing strains that harbor more than one type of toxin gene. So toxins that are produced in higher quantities or that have a higher activity are typically denoted as an uppercase letter, and then toxins that are produced in lower quantities or are less active are denoted with a lowercase letter. So I'm providing all this information to show the complexity of picking a surrogate to represent all the different groups of Clostridium or neurotoxigenic Clostridia, as well as to represent, different toxin types as well. So the botulinum neurotoxins are the most potent natural toxins known. The median lethal dose for a mouse is 0.3 nanograms per kilogram, but the median oral lethal dose for humans is estimated to be around 0.2 to 2 micrograms per kilogram body weight. The toxin is produced as a toxin complex in which it's associated with other non-toxic proteins. And the botulinum neurotoxin is synthesized as a single polypeptide chain of about 150 kilodaltons that is composed of a light chain, which is the actual catalytic domain that causes the enzymatic activity, which I'll describe in the next slide, but also has the heavy chain, which contains the cell receptors binding domain and the translocation domain. So this figure, it just shows the activity of the toxin. So as the toxin is absorbed by the host through either ingestion, injection, or inhalation, the toxin migrates to its target site, which is the peripheral neuromuscular junction, and it binds to the neuron. And once it binds, it's internalized through receptor-mediated endocytosis. And as the pH inside the endosome decreases, the heavy chain undergoes a conformational change, and then that creates a pore channel, allowing the light chain to pass into the neuron cytosol. And the light chain, again, the catalytic domain, will act on the substrate, which are these snare proteins. And the snare proteins are membrane fusion proteins, and they're responsible for the release of acetylcholine at the neuromuscular junction. So if these snare proteins get cleaved, that blocks the neurotransmitter release, and then the muscle cannot function, so it can't contract. So you get and end up with a paralysis of the muscle. So these serotypes can cleave specific snare proteins at specific sites. So they're unique to each different serotype. So now that we have a little bit of information on the organism and how complex and how diverse the organism is and how many toxins are formed and how the toxin works, we're going to talk a little bit more about surrogates. So surrogates are defined by the FDA as referring to a non-pathogenic species and strain responding to a particular treatment in a manner equivalent to a pathogenic species and strain. And there are several criteria that should be considered for selecting an appropriate surrogate, and I've listed them here. So most importantly, the surrogate should be non-pathogenic or non-toxigenic and will not cause disease in humans. And this is especially challenging for selecting a Clostridium botulinum surrogate because in growth inhibition challenge studies, toxicity is what we are testing for rather than growth and determining the success or failure of the food challenge study. So of course, the behavior and activation characteristics and kinetics can be used to predict those of the target microbe when exposed to similar or specific environmental conditions is also very, very important. Clostridium sporogenes PA3679 is a prime example, which has been used as a thermal inactivation surrogate for proteolytic C. botulinum. So that's the group 1 C. botulinum strains. And it's been used as a thermal surrogate for the past 95 years due to its increased heat resistance. So our laboratory here at FRI is currently investigating whether PA3679 and other clostridial strains, closely related Clostridia, grow similarly to proteolytic C. botulinum in foods with and without supplementation with conventional and clean label antimicrobials. I'm not going to actually present any of that data today because we're still generating the data, but I just wanted to point out that's one of our focus, or one focus of our lab. Not only should these surrogates have the same behavior though, and inactivation characteristics and kinetics to predict the target pathogen, but susceptibility to injury should also be similar to that of the target pathogen. So in addition, the surrogate should be simple to prepare and handle and genetically stable. And the genetic stability is an important aspect, especially for surrogates that have been constructed using genetic tools and techniques. And we'll talk about this in a little bit more detail later on, because some of the proposed surrogates, as I mentioned, they've used genetic tools to create gene deletions or gene inactivations. So we'll talk about that. So food products also in general represent a complex matrix that are not sterile. So being able to differentiate the surrogate then from the background microbiota is also key. So providing a selectable marker to distinguish the two or distinguish the surrogate from the background is key. So why do we need a surrogate for Clostridium botulinum? Well, safety is the primary reason we need a surrogate, because C. botulinum, again, produces the most toxic protein on the planet, and can result in botulism if ingested. So furthermore, it's been used previously and continues to have the potential to be used as a bioweapon. Therefore, the organism and its toxin have been listed as Tier 1 select agents, which requires increased security for the institute that houses the agent and the individuals that handle the select agent, adding additional costs to the institute. As a result, there are very few laboratories in the US that are registered to work safely with Clostridium botulinum and their neurotoxins. The Food Research Institute has historically maintained this research program, as has IFISH. And the success or failure of growth inhibition studies relies on whether botulinum neurotoxins are detected in the food samples. And the primary assay for botulinum neurotoxin detection is the mouse bioassay, which is the gold standard assay required for confirmation of toxicity. In vitro assays can be used to screen food samples, but any samples testing positive for toxin production need to be confirmed with a mouse bioassay. Laboratory animal studies are expensive, but there are also the ethical concerns surrounded by the mouse bioassay. For example, for validating the safety of vegan or non-dairy analogs or meat alternative-based foods. So this ethical concern is another driver yet for the search for a surrogate for Clostridium botulinum. Oops. So one of the main types of C. botulinum challenge studies that we perform are growth inhibition studies. And for growth inhibition studies, that involves adding a cocktail of proteolytic and or non-proteolytic spores to a particular food, formulated in the upper ranges of moisture, pH, and water activity to potentially allow C. botulinum to grow and produce toxin when stored during temperature abuse, such as disaster testing at 86 degrees Fahrenheit, or at more moderate temperature abuse, typically how the consumer is expected to store the food. Our refrigerators, my refrigerator, I don't have a thermometer in my refrigerator, so I'm assuming that my refrigerator is probably higher than 4 degrees. And then samples are pulled during the specified time intervals and examined for toxicity, again, using the mouse bioassay. But remember that a good surrogate is non-pathogenic or non-toxigenic, right? So how do we use a non-toxigenic surrogate when toxicity is the endpoint? So this would require construction of a non-toxigenic mutant that you could maybe test a reporter. So that's another avenue that could be used, but that needs to be developed. This is a huge hurdle and probably one of the main reasons why we don't have a surrogate for C. botulinum growth studies. However, if we can correlate the growth of a surrogate through enumeration on plate counts to toxicity, then we could potentially use a surrogate. For the non-proteolytic strains, though, enumeration through direct plating has been very, very challenging, and greater than 1 log decrease in plate counts in comparison with NPN enumeration is typical. And I've listed here some potential surrogates as suggested in the literature, and we'll discuss some of these in more detail in the coming slide. Among the potential surrogates for group 1 C. botulinum includes toxin mutants LNTO1 and the Clostridium mutant for 62A, which I've mentioned, that was developed at UW-Madison. Other closely related species, such as strains of Clostridium sporogenes, tepidum, and tyrobutyricum, are of interest, and this is what our laboratory is actually studying, the use of these 3 organisms as potential surrogates in model meat systems of antimicrobials. Group 2 strains, these are the non-proteolytic strains, often carry their toxin genes on small 48 kb plasmids, and these plasmids have been found to be naturally unstable and are often lost during subculturing. The use of these non-toxigenic group 2 strains then is of interest as surrogates for growth inhibition studies. However, the remaining genes on those plasmids are unknown, and what role they may play in the physiology of the strain remains to be determined. Some research groups have generated non-toxigenic mutants and have proposed their use as potential surrogates for challenge studies. It's unclear why they would choose a non-toxic E strain, why they would generate a non-toxic E strain, because these non-toxigenic E strains are typically the less heat resistant out of the non-protes. So they're kind of like the wimpier strains, the weaker strains. But so the non-proteolytic serotype B strains are underrepresented. The other primary challenge studies that are performed for Clostridium botulinum are the inactivation studies. These studies are conducted to evaluate whether a food formulation or a process or a combination of those can inactivate spores of Clostridium botulinum. So these could include thermal and non-thermal processes that are being evaluated. Low acid canned foods are foods that have a pH of greater than a 4.6 and are packaged in hermetically sealed containers and are processed at time temperature combinations to deliver a 12 log reduction in heat resistant spores of Clostridium botulinum, called the botulinal cook. Clostridium sporogenes is the validated thermal surrogate for group 1 proteolytic C botulinum, and many extended shelf life refrigerated ready to eat foods receive a traditional non-prot bot cook, which is 90 degrees C for 10 minutes or equivalent. And this is to provide a 6 log reduction of non-prot Clostridium botulinum. But spores of Bacillus cereus have been reported to be more heat resistant than the non-prot Clostridium botulinum and can grow at refrigerated temperatures. Some strains can grow at refrigeration temperatures. And so they have been suggested as a target pathogen over the non-proteolytic C botulinum strains. And there's a potential then for these strains to be used, although they are pathogenic, they're not tier 1 select agents. So more laboratories could use these strains. And actually, we've used B. cereus as sort of like a screen for screening lots of different formulations with lots of different antimicrobials prior to validating with a Clostridium botulinum. So the final validation for a challenge study would have to be performed with Clostridium botulinum. So now I'd like to begin to present information on the different surrogates that are currently available and point out the advantages and disadvantages with each one. So the first one is the putrefactive anaerobe Clostridium sporogenes, PA3679. This has historically been used as the model thermal surrogate for the past 95 years and has been used in validating thermal processing technologies for low acid canned foods due to its increased heat resistance. Which is presented here in a comparison of the D values of the strains. And as you can see, PA3679 is much more heat resistant than the group 1C botulinum. And since this strain was identified nearly a century ago, it's been maintained in numerous laboratories throughout the world, and the genome has been sequenced in 2012. So when I was with FDA, we were performing pulsed field gel electrophoresis and whole genome sequencing on all of our C. botulinum and C. sporogenes strains. And as part of that work, we obtained several different isolates of PA3679 from a variety of sources, including ATCC, Campbell Soup Company, the FDA, National Food Laboratory, NFL at the time, and UW-Madison. The ATCC culture we obtained was dissimilar to the PA3679 isolate. So we contacted ATCC. They provided us with 3 spore crops listed as 1961, 1990, and 2007. And through PFG and whole genome sequencing, we found that the 1961 crop was actually contaminated. And the subsequent lots of 1990 and 2007 were Clostridium sporogenes strains, but they were not PA3679. So we were able to isolate the PA3679, which was designated as 1961-4, shown down here. And then we did single nucleotide polymorphism profiling or SNP profiling of the sequence C. sporogenes isolates against a reference strain. And the reference strain was ATCC 15579, and that's what's illustrated here. So the SNP densities, that is the SNPs per kb, in the inner concentric circles are color-coded in this heat map according to a bluish gradient, with the lightest for 0 and the darkest for 200. The cladogram in the middle illustrates the relationships inferred between the various strains. The strains were separated into 2 clades. Clade 1 contained the ATCC isolates, and then clade 2 contained the remaining PA3679 isolates, which included the UW, NFL, FDA, and Campbell Soup isolates. Next, we constructed a maximum likelihood tree of Clostridia 16S ribosomal RNA gene sequences, and the isolates analyzed in that study are shown in light orange, and all were included among group 1 C. botulinum. However, 1961-2, 1990, and 2007 were clustered within a tight group of other C. sporogenes strains, including the strain PA3679 that had previously been sequenced and deposited into GenBank in 2012. So this was kind of alarming. We thought we didn't have the right one. So the remaining C. sporogenes PA3679 isolates from the FDA, UW, NFL, Campbell Soup Company, and 1961-4 were not included in that group, but instead were clustered with other group 1 C. botulinum isolates. So this suggested that PA3679 isn't actually a C. sporogenes, but a C. botulinum that lacks the toxin gene cluster. So we next analyzed the heat resistance of the selected C. sporogenes strains by heating up spore aliquots of each of these of each strain at the various temperatures for about 5 minutes in an oil bath, and then inoculating into duplicate tubes of TPGY broth, which is common growth medium for Clostridium botulinum. And as you can see from this table, clade 1 isolates 1961-2 and 2007 were much more sensitive to high temperature and thus are less heat resistant than the clade 2 isolates 1961-4, FDA, NFL, and UW. And based on this thermal data, we concluded that the clade 2 isolates, FDA, NFL, UW, 1961-4, are more accurately representing the original PA3679 that E.J. Cameron isolated back in 1927. So, and what this also illustrated was that not all C. sporogenes isolates exhibit the same degree of heat resistance as PA3679. And since we have the sequences to decide, we decided to go ahead and look at the genes that may confer this increased heat resistance trait. So, and I want to point out too that, not all sporogenes strains are the same. And I've seen papers that come out that say, hey, we sequenced this sporogenes strains, it's a surrogate for C. botulinum. Well, it might not be a surrogate for botulinum, but you have to test it out or see if the heat resistance is. So the thermal resistance of Clostridium botulinum spores is due to the high content of dipicolinic acid with calcium ions and the uptake of DPA into the spore core. It's regulated by an operon, which is called the SPO5A operon. A mobilizable SPO5A2 operon, which was found to be located on a transposon, was associated in bacillus with increased DPA content and increased spore heat resistance. So we had identified that in PA3679, there were 2 SPO5A operons. And so we attributed the increased heat resistance of that strain to the presence of that extra operon. Blast searches of the contiguous SPO5A2 operon gave high sequence homology to the C. botulinum strains listed here, as well as other Clostridia. And in fact, a recent article by Brunt et al. Out of the UK, they found that 56% of the C. botulinum strains have this second SPO5A operon, but that only 3% of C. sporogenes lineage contain this second SPO5A operon. So when we were looking at this, the ATCC 15579 strain and the clade 1 2007 strain, they lack this second SPO5A operon. And the fact that several group 1 C. botulinum strains contain the second SPO5A2 operon seems paradoxical, as C. botulinum is generally considered to have a lower spore heat resistance than C. sporogenes. In addition, the heat resistance of these particular C. botulinum strains possessing the SPO5A2 locus is not well understood. So it's possible that these strains also feature increased heat resistance, but future studies are needed to actually explore other spore heat resistance factors that may differ between C. botulinum and C. sporogenes. And it would be interesting to look at some of these isolates and whether or not they confer an increased heat resistance as well. So other potential surrogates that have been investigated include a transposon TN916 mutant of the C. botulinum strain 62A. So the transposon TN916, it introduces random mutations and encodes for the tetracycline resistance marker. And that provides a selectable marker to separate it easily from the background microbiota. Remember, that's one of the criteria of selecting a surrogate, is to be able to distinguish it from its background microbiota. So shown in the figure above is the toxin gene cluster arrangement for C. botulinum strain 62A. So there's a toxin gene, and then it's associated with these other non-toxic proteins, the hemagglutinins, and this NTNH, which is the non-toxic non-hemagglutinin protein. So the toxin gene and the genes encoding these associated proteins are here in this cluster, and they are replaced in the LNTO1 mutant with this TN916 transposon. So growth of LMTO1, this mutant, the transposon TN916 mutant of 62A, was compared with PA3679 and C. perfringens in a cooked ground beef model. And PA3679 showed higher growth and a shorter lag phase than LMTO1, but still was comparable in growth. The other mutant that's available is the Clostridium 62A strain. The Clostridium mutant that I had mentioned previously that we worked on when I was a student at UW-Madison. And the Clostridium mutagenesis system introduces a site-specific inactivation within the serotype A gene, the toxin gene of 62A. So this system uses a group 2 intron that contains a nested group 1 intron. And upon insertion of the group 2 intron into the target site, the group 1 intron is excised and restores the integrity of an erythromycin resistance gene, providing, again, another selectable marker to differentiate the strain from background microbiota. Because this is a site-specific gene inactivation, the toxin gene is still present within the genome. It's just simply interrupted. And although the Clostridium system is reported to be stable mutation, there does exist the possibility that the group 1 intron could be excised and the integrity of the toxin gene could then be restored, reverting back to a toxigenic strain. So the stability of the mutant, of the mutation rather, is of concern. And so when we talk about genetic stability of generating a surrogate, that's what we're talking about. So because of that, this strain has not been widely adopted or used as a potential surrogate for growth inhibition challenge studies. Nevertheless, the growth and sporulation of this mutant was comparable in culture media to the wild type 62A strain, demonstrating that inactivation of the toxin gene itself may not negatively impact the growth and sporulation. So moving on to surrogates for the non-proteolytic group 2 strains. So these are the strains that are, again, capable of growth at refrigeration temperatures, but have a lower heat resistance with respect to their spores. Those are the two main differences between these group 2 strains and the group 1 strains. In the early 1990s, three non-toxigenic Clostridium species that were similar, they were just termed non-toxigenic Clostridium species because they didn't know if they were botulinum, but they are very similar to group 2 non-proteolytic Clostridium botulinum. They were identified, and I've listed them here: CMCC 3676, 77, and 78. Analysis of the growth of these strains was compared to 14 non-proteolytic strains at 10 degrees C and revealed that the surrogate cocktail of the 3676 and the 3678 grew equally or faster than the toxigenic non-proteolytic strains, except when the water activity was high, when it was like 0.98 and a pH of 5.5. So we're pushing the boundary. This is illustrated the table also to the left here, which is showing that black or white rectangles represent equal or faster growth of the surrogate in comparison to the toxigenic strains, these 14 different toxigenic strains. This was not the case for the strain CMCC 3677, which is not shown. And the similar results were observed for the surrogate cocktail of the 2 strains than when grown at 7 degrees C, except again at worst case scenarios. Another research group generated 2 mutants in a non-proteolytic serotype E strain, NCTC 11219. One strain is a Clostridium site-specific mutant, and the other was a double homologous recombination, which replaced the type E toxin gene with an erythromycin-resistant gene cassette. So growth and sporulation of these mutants was compared with the wild type strains under stressed and unstressed conditions and were found to be similar to the wild type. No differences were really observed in the D values at 90 and 93 degrees C between the three strains. However, the spore heat resistance was slightly lower for both mutants when heated at 70 degrees C. Growth of the deletion mutant in 2.3% sodium chloride was also slightly faster than the other strains. And it's interesting that the researchers, again, chose to create a non-toxigenic mutant of serotype E strains, since the serotype E strains are much more sensitive to thermal treatments than the non-proteolytic serotype B and F strains. That's what we typically find. So that is, that kind of concludes my presentation, but I hope I presented clearly the definite need for surrogates for group 1 C, for group 1 proteolytic C. botulinum and group 2 non-proteolytic C. botulinum for use in food challenge studies. And the research so far has been devoted to studying strains of related non-toxigenic species or constructing non-toxigenic genetic mutants. The use of non-toxigenic surrogates is vital for the safe handling of the organism and for their potential use in processing facilities and non-select agent laboratories. However, that presents an interesting challenge when the endpoint is toxicity. This challenge may be overcome, again, provided side-by-side comparison studies are performed between surrogate candidates and C. botulinum, and correlations can be made between growth and enumeration versus toxicity. Challenge studies currently use a spore cocktail consisting of 10 group 1 strains, typically 5 type A and 5 type B strains. And yet the only toxigenic mutants that have been generated are non-toxigenic versions of 62A. Or in the case of group 2 strains, it's a serotype E strains. And the group B and F strains are not accurately represented. So we know that the thermal surrogate for group 1 C. botulinum is 3679, which is formerly recognized as C. sporogenes, but has now been more accurately identified as C. botulinum. But again, not all sporogenes strains are as heat resistant as PA 3679, and therefore they may or may not be suitable surrogates for food challenge studies, simply because they're genetically related to C. botulinum. So it's important to consider the growth characteristics and the physiology of the individual strains. So with that, I will open it up to questions. So I thank you for providing the time for me to come and talk with you today. But this is our Applied Food Safety Laboratory. This is the team, because it always takes a team. So they work very hard. So does anybody have any questions?
Great, thank you, Kristin. I'm sure there's a lot of questions. There's many microbiologists I see in this audience. So who wants to go first? Bara.
Yeah, hi. Hi, Kristin. This is a really informative presentation.
Good.
It's great, yeah. And I have one question. Did you try the proteomics or transcriptomics studies along with this finding of SNP in your studies?
The use of proteomics for the studying of what? SMP?
The use of proteomics, like, if you find any of the SNP differentiation between these strains, whether the proteomics are being studied for the finding the toxin is present or not, or something like that? No,
we haven't done any proteomics. I think that would be, I would love to do proteomics for a variety of other reasons. But yes, it would be great to actually tie in to do proteomics. The only proteomics we would do is looking specifically at the protein of botulinum neurotoxins. Okay. So, and we do that by conducting mouse bioassays. And a long history on why, and a long drawn out explanation as to why we conduct mouse bioassays. So the FDA, and a guy can talk on this too, but they will allow the in vitro assays for screening of a bunch of different samples, right? For toxins, for the presence of toxins in foods. But if there are any positives, they have to be confirmed with the mouse bioassay. Well, since we're doing a lot of challenge studies and we really want to make sure that a negative is a negative, we also want to do the mouse bioassay because it's sensitive It tracks all 4 stages of toxin intoxication, of botulinum neurotoxin intoxication. So that's why we do the mouse bioassay. But we can use things like the digital ELISA assay to look specifically for the toxin.
Yeah.
For generating non-toxigenic mutants, you would have to, there have been ideas where people have proposed to use reporter gene systems, where instead of the toxin gene, they would insert like a GFP, green fluorescent protein or something, and then they would track that.
Yeah.
But would it be the same? Would it be expressed the same in bot? We don't know. I mean, no one's actually done this. I've heard arguments saying, no, it doesn't work in bot. But there are other things that could be, maybe tracking NTNH, developing an assay to track NTNH production, which is that non-toxic non-hemagglutinin protein that's transcribed with the toxin gene, but just upstream of the toxin gene. So could that be a marker that people could utilize? So I mean, there's lots of ideas, but nothing has come to fruition.
Yeah,
unfortunately.
I think so. Yeah. Okay. Thank you.
Yeah, thank you.
Yeah.
Anybody else have a question for Kristin? I could ask a question, but I'm sure it's going to be completely off the mark.
No,
but I do have, so I have a question. So
yeah,
and this is going to be more clinical. So when somebody has consumed a product that has, the botulinum contamination, how long does it take? What's the time of onset to see some
2 hours,
toxin effects?
Anywhere between 2 hours and 14 days. And so what happens is when someone gets botulism, well, they don't know they have botulism. They just know they have slurred speech, malaise, drooping eyelids. So they go to the doctor and they say, what's going on? So depending on how much toxin they consume, right? It's all based on dose. It can either be two hours or 14 days, I think is what their typical range is. The doctor will make a diagnosis and the samples will be taken, serum, fecal samples, whatever will be taken from the patient and sent off for confirmation of the diagnosis. So the physician's not going to wait for any test results to come back. They're immediately going to, if they diagnose it. There's other things that look like Guillain-Barré syndrome is a differential diagnosis they have to also consider too, because that's probably more common than botulism. But when they look at those different neurological diseases that could be confused with it, they'll rule out and the physician will actually make the diagnosis, collect samples, send it off for confirmation and wait for confirmation. But in the meantime, CDC will dispatch antitoxin and that's what will be administered in a timely fashion to the patients. Because the antitoxin will then sequester all of the toxin molecules that's still circulating in the blood. Once the toxin has actually reached the neuron and is internalized, I think it's all bets are off. It's going to paralyze that particular neuron or that muscle. But if you can sequester all the toxin molecules prior to that, then you've got a better chance of at least reducing the amount of neurons that will be paralyzed, essentially.
Right. Okay, so and that was my question. I didn't know if there was antitoxins available. So it sounds like there are, but it's just a matter of, can you get to the person soon enough?
Correct. And can they make that diagnosis quick enough? There was, I think it was the carrot juice outbreak. There was a couple in Canada and they actually confused it with heavy metal toxicity, I think. And so that diagnosis was delayed considerably. And they ended up being on a respirator. I think one of them actually died. And then the other patient was on a respirator for 18 months or was hospitalized for 18 months. So, I mean, it's a really long time that it takes for the neurons to regenerate and for people to not be paralyzed anymore. And I think that has to do with clearing of the toxin from the neuron, but also regenerating of the neuromuscular nerve endings. But
okay, well, I have one more question while somebody else thinks of a question. So what have we learned with, with the cosmetic use of botulinum neurotoxin? Has there been any revelations through that research that's helped the food industry?
That's a good question. I don't know exactly. They use cell-based assays for toxin detection because they're looking for potency testing. So they do their potency assays and they'll use probably the mouse or if they don't use the mouse bioassay, they'll use cell-based assays that are pretty sensitive.
And so that's not something that's usable in.
We can, no, because with foods, there are so many, so there's carbohydrates, there's fats, there's all these inhibitory
So the
compounds.
matrix issues. Mhm.
It's huge on matrix issues. If you can purify the toxin from the food extract in such a way that it wouldn't be lethal to the cells, then that would work. But I think a lot of trouble they've had is just it kills off the cells, unfortunately.
Yeah, great, thank you.
Yeah.
Any other questions for Kristin? Behzad, please.
Hi, Kristin, great talk. I really enjoyed it.
Thank you.
And I have a couple of questions. These construct mutant strains that you talked about, how recent they are, what methods they have used to create these mutants?
Oh, so the main mutants that I've talked about is the Clostridium site-specific mutant. So there are not a lot of genetic tools that are available for Clostridium botulinum. Probably there's not a lot of labs working on them. But one of the tools that was developed by the University of Nottingham is a Clostridium tool. And they've refined this and made it easier to use than when I used it back in graduate school days. But that just creates that site-specific inactivation. So the toxin gene is still there. It just kind of interrupts the gene so it's not produced. So the protein, the mRNA transcripts aren't made, and the proteins it's not translated into proteins. So the protein's not made. Like you won't get toxin molecules. But because it's only a site-specific inactivation, there is that possibility that that the intron could splice out or be excised, and then the toxin gene would be restored, right? So that's why nobody really wants to use that as their non-toxigenic surrogate. That's the main hurdle. The other hurdle is when you develop a non-toxigenic mutant, you have to go to CDC if you want it to be used outside in other laboratories, and you have to justify it being a non-toxigenic mutant. So you have to show that upon subculturing it 100 times or 1,000 times or whatever, that the genetic mutation is stable. I think the CRISPR-Cas system is more stable and can generate more stable mutants. But I have not seen the CRISPR-Cas system used for proteolytic C botulinum quite yet, but that would be great if we could do that. So we currently are relying on that Clostridium mutant, but we don't even do that. We were looking at some of these non-toxigenic species, and we've only done one trial, actually, one round, looking at some of these non-toxigenic surrogates and figuring out how much growth you can get with the surrogate before you get toxin production with samples that contain Clostridium. A 2-log growth of the surrogate, then that food failed. So that's what we're trying to establish. And we got some data, but it's only from round 1. And I didn't want to present that kind of data. It's not very convincing all that much, but they seem to be working. I mean, they work in such that we would say probably a 2.5 log, but that has to be refined.
Actually, I had CRISPR-Cas9 in mind when I asked that question. Do you know why it hasn't been tried yet?
I think it actually is being tried. I think it just hasn't been. We haven't generated mutants. So Eric Johnson's lab, my old advisor, he retired. Zabina Pellet is now taking over his lab, and they do work on generating these non-toxigenic isolates, but I don't know where they are in their gene deletion yet. The non-toxigenic, the type E strains that I mentioned that were generated, somebody once said, that was a fluke, because they can't seem to replicate it. So they're trying to figure out how it happened. But they got that double homologous recombination to happen for that particular one strain. And then the other one was a Clostridium type E mutant. But Hopefully, if it's in Zavina's lab, hopefully they'll be able to come out with a non-toxigenic mutant. But then again, you're going to have to look at growth of the strains rather than try to pick up toxin, right? So we always look for toxicity in the food. But if you knock it out and you have just the non-toxigenic strain, you have to measure growth. And measuring growth with Clostridium, we use differential Clostridium media, differential reinforced Clostridium media, DRCA, and the colonies will turn black, but sulfite reducing Clostridia will turn black. So it sort of is, but it sort of isn't a perfect media. Like there's a lot of different differential and selective augurs that are out there for other pathogens, like XLD for salmonella. But for the protes, we have DRCA. For the non-protes, the enumerations are just really bizarre. So we typically get lower counts on plates than we do with MPNs. But to do MPNs on everything is just, you're talking about thousands of tubes, thousands of test tubes. Ask Travis and Guy about the thousands of test tubes. But
Kristin, I have a question about the funding, if I may.
Yeah.
Where do you get your funding for food challenge projects that you pick up? The industry chips in? Or?
Yep. So industry is the primary. We apply for grants just like any other academic lab through USDA, NIH, all that kind of stuff. But most of the projects and the work that we do are brought in through contract projects. So companies will say, hey, we have this new formulation. Usually it's processed cheese. It's Wisconsin. We do a lot of cheese work here. So they'll send it up to us and say, listen, we want to see if this formulation is safe. And so we will set up a food challenge study to assess whether or not that food product is safe. And depending on what the target pathogen would be, we would select whatever target pathogen, and that's what we test. So it's not always just botulism studies. So a lot of studies we do are bot, but we do challenge studies with Bacillus cereus, staph, listeria, whatever would be the target pathogen for that particular food product. Yep.
Thank you so much.
Yeah, sure. Great to see you.
You too.
Uh-oh, Britt, you're muted.
Sure am. Okay, well, I just wanted to say thank you, Kristin, for joining us and really giving an enlightening presentation. I know everybody enjoyed it. I certainly did, learn something new.
Great.
But we're at the top of the hour, and I'm sure everybody has their 2 o'clocks that they need to get to. So thank you again, and hope to see you soon, Kristin.
Yep, thank you.
Okay.
Thank you, Kristin.
Yeah,
Bye-bye.
Thank you.